So essentially what had happened is at the stationary phase, the cells had reached carrying capacity. So they're hanging out, just kind of chilling, not a lot of growth at this point. And essentially what happens with bacteria in a closed environment is that they've reached carrying capacity and then they die off. So there's this death phase. Now, do all the cells die in the culture? No, there will be some in that culture. So let's say that broth culture that you made me, I needed to use it for next semester's microbiology lab. So I go in, I'm like, oh shoot, I didn't order new cultures from the bacterial supply company, wards. And so instead I'm like, hey, I'm going to go ahead and give this a shot. So I might then take some of that sample in the broth and then put it on an agar plate or put it on another slant or put it in another broth. And maybe it takes a while, like it's in a lag phase for a long time. Maybe eventually there's enough cells that essentially survived the death phase and then also this inoculation of a new tube. But essentially, most of those cells are going to die back. And you see that is this gray part in this test tube. So if you want to see some slides, you can pause this presentation, pause my video. But here we go. And then lastly, now there is something that happens when these bacterial cells die. They actually release toxins. So sometimes death can occur quickly because of this toxicity, but sometimes it doesn't. It just depends. So it says their speed with which death occurs depends on the resistance of species and how toxic the conditions are. In my nutrient broth, you wouldn't expect toxic conditions because I created conditions so I actually wanted to grow that bacteria. Okay, practical importance of the growth curve. Well, the very first thing I'm thinking about, without looking at all this on the slide, is when we get sick with a bacterial infection and we're at the doctor and they're essentially asking us lots of questions and then trying to determine, whether we've been whether we have a gram-positive or gram-negative, then thinking about broad spectrum antibiotics and the narrow type of antibiotics or really specific antibiotics. But then they tell you how to take the antibiotic. Okay, pretty important. If they tell you to take it every 12 hours, then take it every 12 hours. Don't do it 8 hours and then wait another 14 hours and then do that kind of thing. So essentially what we're trying to do with antibiotics is we're trying to kill or knock down that bacterial population. And it's going to take maybe 14 days. Maybe it takes 3 days. But essentially thinking about the doubling time, antibiotics can be given to ensure that they are knocking down that population most efficiently. You don't want to take any more antibiotics than you have to, right? Either it's number of days more or more times a day. So we see that in reality, populations can exhibit phases of rapid growth, slow growth, and death. So that's just part of that life cycle that we saw before. So we want to control microbial populations when they're in that exponential growth phase because that's when we're going to kill them off more rapidly. They are in that rapid rate of growth. Once they get into the stationary phase, not so much. So, certainly we can continue to take antibiotics, but we see that essentially it's just easier to start with them when they first start off. Now, thinking about bacterial infections as well, if you think you might be sick and you're having a fever, right, and it's been a couple of days, you probably should get checked out. Because what happens with bacterial infections is that bacteria can migrate kind of like cancer migrates. And if that bacteria is in your blood, then now you have a bacterial infection, which is called sepsis. Sepsis is harder to treat. Now the bacteria is essentially all over your body, not just confined to maybe, the lower left lobe of your lung. So things to think about when you're thinking about the control of bacterial populations. So let's take a minute to talk about how viruses multiply. So we talked about this before a little bit, But what we know about viruses is that they are obnoxious, right? They don't obey the rules of life as we know them, and they are highly specific to taking over and controlling the genetic machinery of cells. So they're essentially using that machinery inside the cell to do all the work for it. So the virus can dictate essentially how it's transmitted, what it does to the host. We see that the responses of the immune defenses are going to be specific to that particular virus. And of course, thinking again about how do we control viruses that are multiplying, then we got to figure out how we're going to control viral infections. So let's take a look. So top right-hand side. So this is the life cycle of animal viruses. So when we think about that virus, essentially what's going to happen is it is going to attach to a host cell. Remember, that's highly specific. I have 3 examples here. Hepatitis B is very restricted into the cell that it attaches to, and that's to the liver cells of humans. Poliovirus kind of has some flexibility, if you will. It prefers intestinal cells, but also nerve cells of primates. And then lastly, rabies is essentially a broad, it's broad in its preferences for host cells. So it can infect various cells of mammals. So take a look. You might remember that our virus has those envelope spikes. And so it's these envelope spikes that are responding to receptors on the host cell. So this is the way it is being specific. So once it attaches to that cell membrane, then essentially we see what we call penetration or uncoating. So that's where this outside this outside coating, as well as those spikes, can become incorporated directly into that plasma membrane of its host cell, or other times it's taken off once it gets into the cytoplasm. I find that pictures are far more explanatory than a bunch of words. So what we see here, again, is that virus cell with its envelope and its spikes. And you can see that it got incorporated by what we call phagocytosis. So it gets incorporated into that host cell. And now you see that the host cell actually has membrane around it. So we call that a vacuole. But essentially that disintegrates. And then you see that the virus opens up, and it essentially is going to release its DNA into the cytoplasm of this host cell. Now, we didn't talk about DNA viruses, RNA viruses, and retroviruses, but essentially what happens next is dependent on what kind of virus it is. I'll leave you to read up more on that if you'd like. So once we see inside the cell, you see assembly. So I should back up. Number 3, we're going to synthesize more of our DNA by hijacking that genetic machinery of the cell. Or if this is RNA, then we'd synthesize more RNA. But essentially what you see is these strands, new strands of genetic material, then are going to get encapsulated by more capsomeres, which create that outside of our virus. And then when this virus is ready, icosahedral here, when it's ready to essentially leave the cell, then it's going to become incorporated again in this plasma membrane from that host cell. And you can see that it's going to get a nice coating of, I believe those are spikes, looking closely, from that host cell. Or I think it's spikes, but also there could be receptors. So there it is. Now it's ejected and it's out doing its thing. Virus life cycles are pretty quick. They don't have much to do, right? Binary fission, you've got to come up with some basic building blocks. For the virus, it essentially is injecting itself into a cell and then using your cellular machinery, making what it needs, and then reusing what it can, and then synthesizing that new genetic material. So DNA viruses essentially enter the host cell's nucleus, where essentially they are replicating DNA. So it's replicated and assembled in the nucleus because that's where DNA is at. RNA viruses, essentially RNA is going to be replicated and assembled in the cytoplasm. That is the same for eukaryotic cells. So the RNA hangs out in the cytoplasm. Now, retroviruses, you see, have a more complex life cycle, and it involves first copying their RNA genomes to make DNA, and then they use that to copy themselves. So anyway, different ways, but either way, they're very good at what they do. I'm going to let you all pause if you want to write down the notes from this slide, but essentially I highlighted this in the one fantastic figure from your textbook. Okay. So this goes on to talk about maturation and release of enveloped viruses. I'm not saying that we need to know this, so let's just skip that. Okay, so let's talk about some viral infections. So we categorize them as acute. You might know that word. That means short-lived, short-term, comes on, and then with proper treatment, it can get cleared up. So for many of us, you might get a cold, and the proper treatment would be drinking plenty of fluids, get some rest, treat your symptoms, but viral infections tend to run its course relatively quickly. So these are acute infections. Some other acute infections, of course, flu, herpes, warts, and chickenpox. So we'll come back to talk about chickenpox. But in our figures down below on the left-hand side, we have a child with measles. Now, measles can be acute and often are acute. So we could talk about acute measles infections, and we're going to talk in a little bit about what happens when this becomes a chronic continuous infection. Okay, right-hand side, we see a child with chickenpox. So in a later PowerPoint, I will tell you how we grow viruses in a lab and make vaccines. But essentially, we know that vaccines are both for bacteria and viruses, depends on the one, but they shorten your symptoms or dramatically reduce any signs or symptoms that you even have been exposed. And it also includes some things there that are more tropical viral infections, including dengue fever, Rift Valley fever, and yellow fever. All right, so persistent viral infection. So essentially, a persistent viral infection is when a virus continues to hang out within its host. So your cells have what they call a carrier relationship with the virus, and you can essentially carry that virus from a few weeks to your entire lifetime. Depending on the virus, it's going to remain latent or dormant somewhere in the body. It's often hanging out in the cytoplasm of a cell. For chickenpox virus, it can reemerge as shingles, so we can designate them as herpes varicella, which is chickenpox, and herpes zoster. So what happens if you've had chickenpox, which I've had chickenpox, is essentially that virus is remaining in this kind of latent or dormant state in your nerves and spinal cord and can become reactivated. So when we think about latent infections, we're thinking about them being reactivated. If you have herpes of the lips, herpes of the genitals, then you know that that's the same way. It can come and go. So you can feel a little tingling and go, oh gosh, like about to have a cold sore develop. And essentially that virus is just coming and going or staying dormant or latent and then becoming active. Okay, measles is on the rise, and let's talk about measles a little bit. When measles is contracted before the age of 2, really the critical time is by the age of 15 months. It turns out that this virus can remain for years. 7 to 10 years after the initial infection, it can essentially be residing in the neurons of the body and can become reactivated, turning into what we call subacute sclerosing panencephalitis. So you can see the stages here, really a terrible condition.